The overall goal of this procedure is to measure changes in vascular permeability to quantify localized allergic responses. This is accomplished by first sensitizing the animals to an allergen, such as ovalbumin. The second step is to challenge the ears with an intradermal injection of the allergen. Then Evans blue dye is injected into the tail vein. The final step is to extract the dye from the challenged ears by incubating them overnight in formamide. Ultimately, spectrophotometry is used to quantify the amount of dye that entered the ear.
The main advantage of this technique over existing methods is that dye extravasation is measured by spectrophotometry. This means that the assay is not easily subjected to observer bias.
Generally, individuals new to this method will struggle because intradermal and tail vein injections are technically challenging and require a lot of practice to achieve proficiency.
Begin with bringing an aliquot of ova stock solution to room temperature for use. When at room temperature, dilute the ova stock to a 1 mg per milliliter concentration in 1x PBS. Use a 5 ml polystyrene round-bottom tube. Vortex the tube at medium speed and carefully add an equal volume of thoroughly homogenized Imject alum to the ova solution dropwise. In parallel, prepare a negative control solution of alum mixed into PBS at a 1:1 ratio using the same technique. Then cap the tubes and secure them to the vortex. Run the vortex at the lowest setting for half an hour with the tubes attached.